So thank you very much for having me. It's really my great pleasure to be here and present this work, which is joint with my excellent collaborator, Ryan Hill, who is a postdoc at Northwestern Kellogg. So today I'll be talking to you about our joint work, Race to the Bottom, Competition and Quality in Science. So basic science is scientific research that is concerned with better understanding our world, but the research itself may not be directly commercializable. However, economists have long understood that basic scientific research serves as a key input into more applied technology and is therefore an important driver of economic growth. And I think that Dr. Vannevar Bush, who led the US Office of Scientific Research and Development during World War II, said it very nicely when he said, "Basic science creates the fund from which the practical applications of knowledge must be drawn." However, precisely because advances in basic science are not marketable, the incentives in this field are different than in more applied research and development. And in particular, we think that credit for new ideas rather than profit is an important currency of scientific careers. And because credit comes, at least in part, from disclosing one's findings first, it's not surprising that we see scientists engaged in these fierce priority races and disputes, each wanting to say that they were the one who got there first. And so on the bottom of this slide, I'm just showing you some perhaps familiar faces. On the left, you see Newton and Leibniz, who fought very famously and bitterly over who ought to be credited with the invention of calculus. On the right, you see Charles Darwin and the lesser known Alfred Wallace, who were both working on a theory of evolution very contemporaneously when Charles Darwin first published his On the Origin of Species. And so when we think about this competition to publish first in science, in many ways, we might think that it's a double-edged sword. On the one hand, we might think that this competition is good for science and society. It might increase the pace of innovation, and it seems likely to induce scientists to disclose their work in a timely manner, because it's only by disclosing your work can you get the credit for having been first. On the other hand, we have heard scientists express concern that this competition to publish first may have a dark side. And in particular, what's going to be the focus of this talk today is this idea that scientists might cut corners and reduce the quality of their work in an effort to publish first. And so actually I want to give you just an example from the field of evolutionary genetics. So Svante Pääbo has this quote that I just think is wonderful because it perfectly lays out the tension we're trying to explore here. So he was working trying to sequence the Neanderthal genome. And as he was doing this, another team was working on the same project. And so this is what he has to say about his competitor, someone named Hendrik. He says, "Hendrik's paper illustrated a dilemma in science. Doing all the analyses and experiments necessary to tell the complete story leaves you vulnerable to being beaten to the press. Even when you publish a better paper, you are seen as mopping up the details after someone who made the real breakthrough." So here you can see the scientist, he's facing this tension between wanting to spend longer to do a more thorough and complete job, but understanding that that opens him up to the risk that somebody's going to come in, preempt him, and publish first. And so the goal of this project really is to answer two related questions. The first is just, is it in fact true that this competition to publish first in science leads to lower quality research? And then second, if yes, what are some of the implications from a welfare and policy perspective? And so we will do this by first just developing a simple model of competition and racing in science. And then what we will do is take the predictions that this model generates and try to test them in a particular field of science known as structural biology. And then at the end, I'll conclude by exploring some of the welfare and policy implications of this priority premium system that we have in science. So I will talk about, yes.
So I have a question. So about this competition on the publication and science and technology, is it a modern thing? I mean, it's more about because we have this established system of publication and peer-reviewed journals, so we have have somehow like a racing to publication, you know. So like in the Newton and Leibniz age, it's somehow different, right? Because they have different systems of publication. So I'm wondering whether your story or model is dependent on this kind of institutional background or not. Yeah.
Yeah, I mean, I think, you know, those examples I gave are hundreds of years old. I think, you know, now it is clear who wins, because we, as you say, we have this system of peer review. The journal articles come out with a date on them. You know, I think the part of the reason that this Newton and Leibniz dispute was so ugly was that it was hard to adjudicate at that time, when people are just sort of publishing. And so, yeah, I would probably say, I guess it's not clear to me though, if it's worse or better in the modern age. You know, in some sense, there's a clear finish line, so maybe people race harder. On the other hand, at least there's less fighting, I think, ex post. Although, you know, on the other end, even that's not clear, I guess, if you look at sort of.
Yeah, in ex ante, there will be more competition,
yeah,
but ex post, you know, you have the dates on the paper, so.
Yeah. Yeah, but I mean, this stuff is always messy. If you think about the patents over this CRISPR gene editing technology, for example, you know, there's a very clean, clear patent system, and yet that didn't stop there from being a very messy dispute between MIT and Harvard and UC Berkeley. So yeah, I don't have a great answer. I'm not enough of a historian, but it's certainly interesting to think about how these systems have evolved over time.
Yeah.
Yeah.
Carolyn,
yes.
I have a related question. So when you say low quality research, it can also be like I may publish more preliminary finding. It means, because you know, for science, it's not like you get the finding like immediately, or the full story. You get you take steps, right?
Yes.
So you may like tell people you have some finding, but not the whole story.
Yes. Yes.
I mean, it's not like they, I don't know, it's not like low quality. It's just like they announce the early finding and
yes. So I think like another way of phrasing your question is, if you think of sort of science or innovation as proceeding like a quality ladder, where sort of people make steps and then other people, or they can build on each other's steps. And if you release early and you sort of just release at an earlier step on the ladder, that may not be bad for science. In fact, that might be good, because then you or somebody else could come and build on it. So what we want to argue that is sort of, I think it's true in this particular field of science, and I think in other fields too, is that when you rush in this field, it's not costless for somebody to come and improve on what you've done. If you've rushed here and let's say you've run an experiment where you produced poor data, at that point, if somebody would like to improve on what you've done, they can't just reanalyze your bad data. They would have to go all the way back to the beginning and start the experiment again. And so if that's the case, then it has been costly. But, you know, another example you might think is like a field of science, like say where, you know, I'm a naturalist and I'm just collecting species of birds, and I can publish the paper when I've cataloged 100 species, or I could release it when I've cataloged 200. I don't think it would be bad for science if I stopped early and just put out the paper with 100, because everyone can use that and just keep adding. But here it's a little bit different, because I think that we've sort of made a mistake that's going to mean that to improve, we have to resync the same costs. And so that's, I think, a really important point. I'll try to sort of return to that when it's relevant in the setting. So yeah, I'm going to focus on this particular field of science called structural biology, and I will talk about it in depth. But just to preview, you know, why this type of science. So first of all, what is structural biology? Structural biology is the study of the 3D structure of biological macromolecules, which for the most part means proteins. And so one reason to study it is just that it's a very important field of science. Over a dozen Nobel Prizes have been awarded in the field. It has a lot of applications in disease and drug discovery. Just to give you a very topical example of why we might care about structural biology, on the right here, you're seeing a clip from Science magazine, and you're seeing a rendering of the 3D structure of the COVID-19 spike protein. So if you've seen what the COVID-19 virus looks like, you've probably seen that it's coated in these little spiky objects. Though these are the spike protein. And it's exactly this protein that your body needs to learn how to recognize in order to mount its immune response. So very early in the pandemic, structural biologists were very focused on figuring out that 3D structure, which in turn has been an important input into the vaccine development efforts. So that's just one example of how this is an important field of science. For our purposes, the data in this field are just uniquely good. And so we take advantage of a free public open access resource known as the Protein Data Bank that's going to give us things like objective measures of the project or the protein structure's quality, fairly detailed project timelines that's going to let us know who was working on what and when, links to the paper that debuts the protein structure, as well as other sort of scientific details about the project. So just to quickly preview the results before diving in more detail, the key prediction that comes from our simple theoretical model is that projects that scientists understand before starting are very important. So a protein that a scientist sitting in his or her lab says, I know if I solve this, this is going to be a blockbuster publication. These are the types of projects that we expect a lot of scientists to be interested in working on. So it means that it's exactly these important projects that are going to be very competitive. But then it's exactly these important projects that we expect to be the most rushed and ultimately done with the poorest quality. The empirical results really support this prediction. So we can see that these projects that we code as being very ex ante important, we call them high potential projects, they're more competitive in the sense that we can see that multiple researchers were working on them contemporaneously. And then we see that these same high potential projects are done on a more compressed timeline and are ultimately done with lower quality. We then look at follow-on work. So I think this is sort of related to this question that came up. Follow-on work in the sense that do other researchers come along and try to fix up or redo this same protein? And we do see that happening. That helps, it ameliorates this negative relationship between the importance and the quality, but it does not totally eliminate the negative relationship. And then lastly, I'll show some suggestive evidence that these quality magnitudes we're talking about are large enough to impact how useful these protein structures are for more applied drug development work. So in other words, we're losing enough quality here that we end up, we think, getting less R&D that builds on top of the science. Lastly, we sort of return to the model to try to say something about the welfare here. So the first thing we can say is that this negative relationship between the importance of the project and the quality is inconsistent with an idealized first best, in the sense that if the social planner could tell researchers which projects to work on and how long to spend working on them, we wouldn't expect to see this. But that may be sort of an unrealistic standard. And so instead, if you just say the social planner, the only lever they have is how to divide credit between the first and second place team. So do we make these races winner take all? Do we make them more even, sharing the credit between the team that's first and second? If that's the only lever you give the social planner, then it may in fact be the case that the best you can do is to have these rewards be unequal. And if that's the case, you were going to have this negative relationship between the importance and the quality of the question. So in other words, this result we find in the data is inconsistent with an idealized first best, but may very well be consistent with a constrained second best. Okay.
Sorry,
so
Carolyn.
Yes.
Yeah. What's your definition of the potential?
Like in sort of like how are we going to measure it empirically?
Yeah, both theoretically and empirically.
Okay. Yeah, so I think theoretically it's the scientist's expectation of the payoff of that project.
Mm-hmm.
So some projects are high payoff because they answer a long-standing open question. Others, are just not very important. So that's sort of what it means theoretically. Empirically, what we do is we use the very detailed scientific characteristics of the protein. So things like what does the protein do? What organism does it come from? to predict how many citations the associated paper is going to get. And that measure of predicted citations is going to be the potential. Yeah.
Okay, so about this this slide before, yeah. So it's basically a story about waste of resources, because even though we have maybe the step of the progression is smaller for each scientist, but maybe we have a lot of scientists comes into this field because of their, just like you say, you're racing to the bottom, but you have more scientists come in because they can just contribute a little bit and get a lot of citations, right? So that's another effect you will consider.
That's another effect. We are sort of, we don't make a statement about whether we have sort of over entry into the important problems. That's possible. It's not obvious, because if there's a risk of failure, you as the social planner would probably tolerate ex post some duplicated effort, because you're minimizing the probability that everybody fails. So I think, just because you see multiple people working on the same project, that alone doesn't tell you that there's sort of over entry.
Yeah.
And
so
our model sort of is not equipped, I think, to sort of make statements about whether there's sort of over entry or under entry in these important problems. I think that's a little distinct question.
Yeah. Thank you.
Yeah, no, that's a good question. And I think, there's other people who've worked on that. Hugo Hopenhagen, I know, has some work on sort of excess entry. And I think, a lot of it has to do with how the adjustment costs. If it's very costly to pivot to a new project, then it's very wasteful to have lots of people working on the same one, because once one person wins, everyone else has to adjust away.
In reality, usually it's because the principal who is providing the funding to the scientists, it's a step-by-step funding. So you have to give some results and review some positive information or signals to the funding manager, and they will continue the funding to you. So I mean, this kind of game may also lead to this kind of racing to the bottom effect, right?
Yes, I think so. So we sort of see this fitting into, I think, a couple distinct literatures. We were very influenced by the work of Robert Merton, the sociologist, who really sort of in the 50s and 60s was one of the first people really thinking hard about the incentives in science and how that governs sort of the production of new knowledge. I think then separately, this work is very related to some of this IO work that thought about strategic behavior and patent and R&D races. And then lastly, I think we're hoping to contribute to sort of this scientific literature, or even sort of more of an op-ed literature, where scientists have just expressed concern that as science grows more competitive, this may not actually be good for the production of new knowledge. And so what we hope our primary contribution can really be is just to try to bring some empirics to what really has largely been a theoretical or speculative discussion. Okay, so let me now briefly talk about the model. I think the empirics are more interesting, so I'll try to sort of balance the time to spend more time there. But let me start by saying that if I had to describe the model to you on one slide with 5 bullets, this is what I would say, and then I'll give a little bit more detail. So we conceive of projects as varying in their ex-ante potential. We denote this P. This is the scientist's expectation of the payoff of the project. Scientists, before they start a project, understand the underlying science. They know sort of the questions the field is interested in. And so I think it's very reasonable to think they have well-calibrated expectations about, how well received a project would be if they were to succeed. That's what we mean when we say potential. Then scientists, once they start working on a project, decide how long to spend working on it. We denote that M for how long are they going to let the project mature for. And when they're making that decision, what they're trading off is, on the one hand, spending longer translates to higher quality, Q of M. They're trading off that increased quality against the threat of being scooped. And the balance of that trade-off is going to dictate when they decide the quality is good enough, I'm going to stop and publish what I have. The key ingredient that really makes this whole model turn is that entry into projects is endogenous. Scientists choose which projects to enter. And what that means is that there's likely to be more competition in these high payoff, high potential projects. The way we actually operationalize this in the model is that we say scientists, if they want to start a project up, need to sink some investment cost I, and that's going to generate for them a probability that they successfully get the project going, G of I. And so if they choose to invest more, there's a higher probability that the project starts. The key result that comes from this is that these high potential projects, because they're more competitive, people have invested more to start them up, these are exactly the projects that are going to be cut off the soonest, the rushed, and ultimately executed with the lowest quality. And the key policy lever we imagine a social planner being able to manipulate is the credit split between the team that publishes first and the team that publishes second. So we call that theta upper bar, that's the credit that goes to the first place team, and theta lower bar, the credit that goes to the second place team. So just to give you a little bit more detail on how the timing works. So here we're looking at scientist little I. She's going to start this project at some time TIS. And that's when she's going to decide how much to invest, this capital I, in hopes of trying to get the project started. So, one way to think about this is how long am I going to spend writing my grant proposal? And the more time and energy I put in, the more likely I get the grant funding, and I can't start until I get the grant funding. In our context, I think we have a more particular view of what this corresponds to, but that's sort of one general rationalization. So she's going to invest capital I and start the project with probability G of I. If she successfully starts the project, that's when she's going to decide how long am I going to spend on it. That's when she's going to choose this little m, and she's going to be trading off the increased quality you get from spending longer against this threat that somebody else is going to publish first. And so in this example, she does publish first, and she's going to get a payoff. It's going to be a function of three things. It's going to be a function of P, the project's potential. It's going to be a function of Q, the quality of the project. And then it's going to be weighted by this theta upper bar because scientist I finished first. Scientist J goes through an exactly analogous timeline, but in this example I'm showing you here, J started second, J finishes second, and so J is going to get a payoff that is identical but weighted by this theta lower bar, the credit share that goes to the second place team. Now, what's very critical for sort of governing how these two scientists are going to behave in this race is what do they know about each other? And so in particular, what does scientist I know about scientist J? So scientist I will know that J entered with probability G of IJ. And in equilibrium, I will have correct beliefs about how much J invests, and therefore the probability that J is in the race. So I does not know if J are in or out, but I does have correct beliefs about the probability. The second thing is that I needs to have some beliefs about when J started. And so we here try to assume something just very simple. We just say that I believes that no matter when they start, they are equally likely to be ahead or behind in the race. And in particular, they're just going to think that J's start time is uniformly distributed around their own start time. And so just to be sort of very transparent about what this buys us, this means that I's start time in terms of calendar time is just not informative about when she's ahead or behind. If I starts in January or I starts in June, she thinks in either case, she's equally likely to be ahead or behind in the race. And so in turn, the strategy, this choice of M, is just not going to depend on calendar time. It's going to be independent of calendar time. That's going to make things much easier for us. And we think sort of in terms of the data we have at our disposal to actually test, we're not really losing any interesting testable predictions by making this simplification.
Carolyn?
Okay, yes.
Yeah, I have a question. So this uniform distribution buys us this simplicity of calendar time doesn't matter, but that's just this scientist J's belief, right? But in the equilibrium, whether the scientists enter the field earlier or not or later may matter a lot or not.
I mean, I think it's plausible to think that I have some sense that I started in July and I'm probably behind, and we're just
No,
not.
no, no, no. I just say in equilibrium in this model. So if I enter the field maybe a little bit late than others, that will affect my investment or anything else, maybe my my achievement, my citations, right?
Well, so the idea is that if you enter late, you don't know that you've entered late.
Yeah,
and
but
so,
in equilibrium, you know, but exposed, you have a result, right?
yes, but we're saying that you sort of commit to M at the start, and
Okay.
you have no conception of whether you're early or late. And so it's just not going to change your behavior.
Yeah, it's not going to change the behavior.
Yeah.
Yes.
Yeah.
Yes.
And in fact, I think that it's not the uniformity that's important. It's the symmetry. It's just the symmetry. Yeah. Okay. And so, you know, that's sort of the setup. And then I think the three predictions that come out of the setup are fairly intuitive. The first is just that high potential projects generate more investment. People choose a larger I if the project is very important. And so then therefore, in expectation, these are the more competitive projects, the projects we expect a race to be more likely. The second proposition is that if there's a project where I think it's very likely that my competitor has indeed entered, it's a competitive project, that's the project where this choice of the optimal M, I'm going to lean towards choosing a shorter M. And so those projects are going to be completed faster and with lower quality. And if you put 1 and 2 together, you get what we see as sort of the key prediction, which is that it's these high potential important projects that are completed faster and with lower quality. And so these are the predictions we're going to try to take to the data. And so now what I'd like to do is just spend a little bit of time telling you some things about structural biology and trying to just be very careful to explain how we're mapping from variables in the model to things that we can actually observe in the data. So as I said in the introduction, structural biology is the study of the 3D molecular structure of proteins. And here I'm just showing you some images. This is sort of what the finished product looks like. And I picked sort of particularly important proteins that just highlight the important applications this field has in medicine and drug discovery. And so how does a scientist actually go about solving a protein structure? So the vast majority of protein structures are solved using a method known as X-ray crystallography, which broadly involves 3 steps. The first step is to purify the protein and then try to grow this protein that's extremely tiny, too small to handle, to try to grow it into a crystal. So if you've ever grown a salt crystal in your kitchen, you sort of have an idea of what we're trying to do here. You create a really saturated solution of the proteins, and then you slowly evaporate the water out, and you hope that the proteins just come together in this nice orderly way. So it's sort of, in principle, that's what you're trying to do. In practice, it just is exceedingly complicated. There's hundreds of different experimental conditions you can tweak, things like the temperature, the pH, other molecules you add to the solution, all of which are, matter for whether you're going to successfully grow a crystal. And so this is the part of the project actually that can be extremely time consuming and very risky. And so we really think of this like the investment part in the model. You can invest a lot in the sense of trying lots of different experimental conditions. That's going to increase the likelihood that you stumble upon something that works. But at the end of the day, this is an uncertain stochastic process. However, once you have a crystal, you take it to a facility known as a synchrotron and you beam it with X-rays. That's going to create something called a diffraction pattern that you can see on the right here, this white square with the little black dots. So that's like your experimental data. And what the scientist tries to do from here is basically reverse engineer what must have been the 3D protein structure that would have created this diffraction pattern. But importantly, this is an iterative process. So the scientist will propose a 3D structure. They will then say, if this was really the structure, what would the diffraction pattern look like? They'll compare that to their experimental diffraction pattern and they won't match. So they'll go back to their model, they'll tweak, and they'll repeat this process. This is known as refinement. So this is really like the maturation stage in the model. The more cycles we do of tweaking and checking, the better and better the protein model will be, until at some point the scientist decides this is good enough, let's write the paper up. Okay, so that's sort of what they're doing. And then what is the protein data bank, this data source we're using? So the protein data bank was established in the early 1970s, and it's a database for these 3D structures. Since the 90s, most scientific journals and funding agencies require scientists to submit their data to the protein data bank. Otherwise, they will not publish their papers. So that means that for our purposes, it's a very complete census of the proteins that have been solved by scientists. And today, the protein data bank contains well over 100,000 protein structures and is growing at a rate of about 10% a year. So if you're curious, you can log on to the protein data bank, and if you search for protein structure 4CMP, this is what you'll see. And so I picked a particularly important protein. This is the CRISPR-Associated protein 9, or the Cas9 protein. It's very important in the CRISPR gene editing process, but all the pages look very similar. And so you'll typically see the rendering of the 3D structure. You'll see the date that the protein was deposited, and then the date that it was made public in the protein data bank. You'll see these measures of the protein structure's quality that I'll talk in detail about. And then you'll see the link to the paper that first discusses this protein structure. And in turn, that lets us see things like how many citations the paper receives. So the first key thing that we can observe, that we have in the model that we'd like to observe in the data, is this idea of the project's quality. And so I just want to say, like, this is very, I think, unique to this field that we can observe this. In economics, if somebody said to me, can you systematically rank papers in terms of their quality, that would be very hard. We could try to say how many citations does the paper get, but that's going to exactly conflate sort of the importance of the question with perhaps how well done the analysis is. And really the point here is to try to separate those two. And so what's very exciting about structural biology is that they have developed these measures of project quality that are really sort of exactly engineered to be what we want, because the protein data bank wants to be a useful resource to scientists in other fields who may not be experts themselves in structural biology. And so they developed these measures so that sort of a novice can look and get a summary statistic that says, how good is this structure and how much confidence should you have in it? So that was sort of the impetus for developing these measures. And so we use 3 distinct measures. The first is the structure's refinement resolution. And you can really think of this as being very similar to just the resolution of a photograph. And so actually what I'm showing you in this image here is the same protein structure, but as you move to the right, the refinement resolution is just improving. And you can see that you can view much more detail when there's a good refinement resolution score. The second measure we have is something known as R-free, that is meant to measure the model fit. And so in other words, it's saying, what is the agreement between the experimental data and the data that would be generated from your proposed model? And actually the protein data bank is pretty savvy. So they realized they were having issues with people overfitting their data. And so now they actually have them reserve a holdout sample of their experimental data, and they assess the model fit on the holdout sample, trying to avoid issues of overfitting. The last measure we have is what we call this outlier measure. So someone will propose a 3D model, and you can look at this and you can look at it bond by bond. And as a chemist, you will know that certain molecules should only bond at certain angles. And so what they do is they count up every time your model has these bonds that we know are chemically implausible, that these 2 molecules should not be bound at this angle. And they count those up and then report them as a fraction of the total bonds in the structure. So you might get something like, a score of 10% of your bonds are sort of failing this sanity check. And so those are the 3 measures we have. And in the paper, we look at all of them separately. What I do in the talk, just to make the exposition a little bit easier, is combine them into a single index. And all the signs will be such that higher values correspond to better quality, and the units will be reported in standard deviations. Okay, The next thing that is sort of an important piece of the model that we can actually observe that is pretty cool is we can actually see to some extent how long scientists spend working on the project. And that's because in the protein data bank, the scientists are required to report key dates. And so the project starts when the scientist tries to grow the protein crystals. And unfortunately, we don't know when that happens. But once they have a crystal in hand and they take it to a synchrotron and beam it with X-rays, that date is recorded. And then we see the date that they deposit the structure in the protein data bank, which they need to do before they can get the structure published. So we can see how much time goes by between when they first get their data and when they ultimately deposit into the protein data bank. So what we think is going on in that time is they're doing this process of building the 3D model from their data and also maybe writing up the paper. And so on average, scientists are spending about 1.5 years doing this, but there's quite a lot of variance. Okay, the next thing we would like to be able to observe is some measure, sort of protein by protein, how competitive was the protein. And that's something that we can't see, because what we really want is the scientist's perception when they're working on the protein of how likely is it that somebody else is working on it. And we can't see that. But what we can see is ex post, how many people deposited the same structure in rapid succession, as sort of a way of trying to proxy for how much competition do we think was going on. And so the way we're able to do that is we can ask the protein data bank to group together all the proteins that have the identical amino acid sequence, which basically means they're exactly the same protein. And then what we do is we just count up the number of times that protein was deposited in the protein data bank within the 2 years of the first structure being deposited. So what we're trying to do here is say, we think that these were instances where people were working contemporaneously. At some point, the protein may continue to get deposited, but people know it's already been solved. They're just choosing to do it again. So we're trying to separate between the two. So this 2-year cutoff is a little bit ad hoc. The results are robust to sort of, if you want to use a 1-year window, a 3-year window, but we're just trying to separate between this protein when it was being worked on contemporaneously versus when other people just ex post decided to redo the project. But what's important to note here is that even if we get sort of this exactly right, it's going to be a noisy proxy for the thing we really want, which is the scientist's expectations that there was somebody else working on it. We're seeing the realization that's going to be a noisy measure of sort of the perceived competition at the time. So I just highlight this because I think this is a fine measure for us to use if we want to put it on the left-hand side as an outcome in a regression. But if we want to put this on the right-hand side, say, you know, how does competition impact quality, for example, we're going to have big issues with attenuation bias. And if I have time, I'll show you some IV results that suggest that that is indeed the case.
Carolyn?
Okay, yes.
So basically you can only observe the exposed successful projects, right?
Yes, yes.
Well, those projects that were failed before, then you cannot observe that, right?
That's
Maybe it's a very hard protein to produce, so you have a huge amounts of entry, but in the end you just have several survivals, right? So
although I don't know if I thought I had a lot of people trying to work on it, but the success rate was going to be very low, I don't know that I would feel that that's a lot of competition. Because even if I knew that 5 other labs, if I'm sort of working on something, making good progress on it, and I think 5 other labs are trying to work on it, but I know it's really hard, I would feel less threatened than if it was a project that I thought was pretty easy and 5 other labs are working on it, because then I would think it's very likely that one of them is going to succeed. So I don't know that that's so much of a problem. It's just more that really what I want to capture is my beliefs about the number of other labs. And we just can't see that. We see this sort of very noisy thing, which is just how many ultimately, even if my belief, even if sort of beliefs are correct, there's still going to be noise. And that's just going to introduce attenuation bias.
Yeah, you're right. So maybe a stupid question. Why people repeat the experiment to produce the same protein?
So I think because some of these very important projects, the first time because of this rushing, are done with bad quality. And so, and I'll show you some results that exactly suggest that people basically make this calculated decision. They say, you know, I'm going to be second, so I'm not, but I can do a much better job. And maybe then it's worth it. And that's going to be very helpful, I think, for science, actually, because it means that ultimately, for the most important proteins, even if people are rushing, eventually we get it okay. Someone comes in and does a good job.
Yeah, so then you can distinguish the competition and the improvements, right?
Mm-hmm. Exactly.
Yeah.
Exactly.
Okay.
Yes. So the last thing that I think the model sort of tells us we need that we can't directly observe is this measure of the project's potential. And so one way you could try to do this is to just say, how many citations does the paper ultimately get? But we think that that's maybe not great. For one thing, it might conflate the project's potential with the quality that it was executed. So we think a cleaner thing to do is try to use really detailed characteristics of the protein to predict how many citations we think the paper will get. And so in some sense, what we're trying to do is recreate the scientist's thought process. The scientist is going to know a lot about the protein, and that's going to inform their beliefs about how important and well-cited this project will be. So just to give you sort of an idea of these types, these covariates we have are very detailed. So in particular, we'll have this pretty detailed classification of what the protein does. Is it a membrane protein, for example? Does it transport oxygen? We'll know the taxonomy or the organism that it comes from. We'll know the gene that it's linked to. We'll even know the number of papers that have been written about this protein before anyone knew the structure of it. So often we'll know a protein exists and scientists will be interested and they'll write papers about this protein, but nobody has yet solved the structure. So that's sort of a very nice measure of the ex-ante demand or interest in this protein structure. So using those covariates, we actually have, a lot. So we use lasso to select the model to avoid overfitting. And we use that to predict the citation percentile we think that this protein structure is going to ultimately be. And so what I'm showing you here in this figure is on the x-axis, the predicted citation percentile, and on the y-axis, the actual citation percentile. And then I'm showing you here a binned scatter plot. So just to be very clear, every dot on the scatter plot is many, many proteins. So not to say that all of the data lies this close to the line. The R squared is actually about 0.2. But just to say that sort of throughout the distribution, the fit appears to be pretty good. And we can explain about 20% of the variance in actual citations using these covariates. And so that predicted value is going to be how we measure the project's potential. So with all these pieces in hand, we can now actually go about trying to test the theory. So the first proposition, if you remember, is that high potential structures ought to be the ones that more scientists are trying to work on. They should be more competitive. And so that's exactly what I'm showing you here. So I'm showing you a binned scatter plot. The regression equation is on the bottom. So on the y-axis, I'm showing you the log number of deposits. So just the log number of times that protein was deposited within 2 years of the first deposit. On the x-axis, I have our measure of the project's potential. And so you can see that there's this positive relationship. The high potential projects look like they are deposited more times in quick succession. And so what I'll do sort of throughout to try to benchmark the effect sizes, just because I think reporting these beta coefficients is not particularly helpful, what I'll do is just report the difference between a protein that falls in the 90th percentile of the potential distribution to a protein that falls in the 10th percentile of the potential distribution. So you can just think of this as like the comparison between a high potential protein and a low potential protein. So if you do this 90-10 comparison here, what this is saying is that the high potential proteins are deposited 30% more times on average than the low potential proteins. Then sort of turning to this sort of what we think of as the key model prediction, the high potential projects ought to be completed faster. So here it's the exact same regression and graph, but now the y-axis is maturation, the years between when the data was collected and when the protein was deposited in the protein data bank. And you can see that these high potential projects are completed more quickly. And so again, doing this 90-10 comparison, it looks like the high potential structures are finished about 0.2 years or just over 2 months faster than their low potential counterparts. And this in turn seems to translate to lower quality. So now we have this standardized quality index on the y-axis. And again, doing this 90-10 comparison, you can see that these high potential structures are about 0.7 standard deviations lower in terms of quality than the low potential structures. Okay, so this is sort of, I think, our main result. But when you look at this, I think something that we were very concerned about is, what about omitted variables bias? And I think in particular, a story we were worried about is, what if the high potential projects are just more complicated? Because that could be a reason that has nothing to do with racing or rushing, that they would be just executed with lower quality. And so that's a concern that we took very seriously. And what I want to do now is show you 3 distinct pieces of evidence that I think push back against that being the explanation for these results.
So, okay, can I ask a question? So, is it possible that because this is a high potential project, so there are more tries, and I think like for the best, for the best one, it's still high quality, but there are many low quality tries, so that will drive down the mean.
Yes, it's like an order statistic problem, is what you're saying, basically. Like that's a good point. And I think, I think the right thing to do there is to like look at the quality of the team that comes second and third, and sort of try to verify that those ones are also low quality. And it's not just that we have sort of cherry picked by focusing on the winner, we've sort of mechanically selected the lowest quality structure. So I can't remember if we've done exactly that. I'm pretty sure that even if we sort of looked at, the second or third one, we would still see this. We see quality improvements coming later. So, 2 years later, for example, somebody will deposit a better version of the structure, because at that point the race is over. And so we think that then people are actually taking their time. But yeah, your point is well taken. And I think, we can actually just try to sort of carefully verify that that's not what's driving this. I mean, I'm interpreting your question correctly, right?
Yeah, and another related question is that, is this what the founders really want? I mean, when we say it's high potential, usually like people want the progress be faster, right? So they want the researchers to get things done faster, even if it's not perfect. So like COVID-19, like the countries put a lot of money to ask people to speed up, right? So that might not be a bad thing, like people.
Yes, and I think when I get to the welfare section, I think this will be a little bit clearer. So the way we've thought of this is that the social planner and the scientist have the same discount rate. And so the scientist, in the absence of competition, would sort of let the project mature as much as the social planner would like. There's sort of no wedge there. Now, I think it is, totally plausible view of the world that actually scientists are sort of too patient relative to the social planner. They don't have the same discount rate. So then in that case, actually, this priority rule is sort of helpful in pushing us back towards a first best, because it can kind of align the discount rates. So I think that that's sort of beyond the scope of our model, but a sort of very reasonable view of the world to hold. Yes. Okay. So with this concern in mind that these high potential projects are more complicated, one thing we can try to do is just control directly for how complicated the proteins are. And I think actually, based on our conversations with scientists, this is something that we can do somewhat successfully. Because when we talk to scientists, one thing that became very clear to us is that this process of crystallizing the structure is very difficult. It's also really difficult for them to predict ahead of time which proteins are going to crystallize easily and which proteins are going to be really hard to crystallize. And so the fact that scientists sort of can't forecast that makes us think it's just pretty uncorrelated with anything. So that's a helpful fact. That being said, larger proteins do tend to be more difficult to crystallize. And moreover, once you've crystallized them and have data, it's a more complicated model to build. So the size of the protein, we think, is a very good proxy for how hard the protein is to solve. And so what we do is just try to control for 3 different measures we have of the protein size. So one is how much does it weigh? One is its residue count, which is the number of amino acids it has. And one is its atom site count, the number of atoms that it has. And so what we do is we just take our regressions, our potential quality regressions, and we add these complexity controls. And so what I'm showing you here are actually 4 regressions. I'm showing you all 3 of the different quality outcomes separately, plus this index. And in panel A, I'm showing you the regression between quality and potential with no complexity controls. And in panel B, I am adding those complexity controls in. And I think 2 things jump out here. The first is that when we add these complexity controls, the R squared goes up a lot. So these controls are powerful in the sense that they really do explain a lot of the quality or lack thereof. That being said, the coefficients on the potential measure are quite stable. So what that says to us is that complexity is very predictive of the protein's ultimate quality, but it's just not very correlated with how important the protein is. So that is a sort of very reassuring fact that suggests that complexity, while it sort of matters for quality, cannot explain this negative relationship between potential and quality. The second thing we can do is just sort of point out the fact that if you take this story seriously, that A, scientists are agnostic toward competition, but B, the high potential structures are more complicated, then you would expect scientists to spend longer on these important but difficult proteins. And in the data, we see the opposite. We see that these high potential structures are completed faster. So that again seems more consistent with the story of rushing than it does with just some projects being harder than others. The last thing that we can do is take advantage of the fact that different scientists in our data face different incentives. And in particular, most of the scientists in our data are scientists who are affiliated with universities, and we think that they're sort of part of this academic publish or perish world. But a meaningful subset of the researchers in our sample are affiliated with these research centers known as structural genomics consortia. And these structural genomics consortia are publicly funded research groups whose stated mission was just to solve as many protein structures as they could and promptly deposit them in the protein data bank to be a helpful public good for other researchers to use. And importantly, we think that these structural genomics researchers are much less focused on publication and priority. So this is a group who really faces less of these competitive publish first type incentives. And in fact, we can actually just observe this directly in the data. So of the structures deposited by university scientists, over 80% of them end up being published in a journal. Whereas for these structural genomics researchers, only about 20% of their structures end up being published in a journal. So we think this is a group of scientists that really is just much less focused on publication. And so what we can do is just compare the behavior of the scientists in these 2 groups and see what happens if we turn off or at least substantially tamp down competition. So what really this is just a difference in differences strategy, and I'm just trying to visualize it for you here on this graph. So again, what we have here is potential on the x-axis, the maturation period on the y-axis, and in blue you're seeing these university scientists, and in red you're seeing these structural genomics researchers. So the first thing that really jumps out is just the level shift in this picture. So it looks like the structural genomics researchers just are getting things done much faster. Now, to us, I think that makes a lot of sense because if you recall, the majority of these structural genomics researchers are not trying to write a paper. So as soon as they have their structure, they just deposit it in the protein data bank. Whereas the academic researchers are also writing a draft and basically waiting until they're late enough in the publication process that they need to provide evidence that they've deposited to the protein data bank. So it's not surprising to us that we see this level shift. What I want to highlight here is the difference in the slopes. So as I showed you before, these in blue, these academic researchers, they spend less time on the more important structures. On the other hand, these structural genomics researchers who we think are less competitive, if anything, they spend more time on the more important structures. So in other words, if you sort of turn competition off, you no longer have it be the case that the important structures are done more quickly. So it seems like that is a fact that arises exactly sort of as our model suggests, because people want to be first and they're very aware that they probably have a competitor. Now we can do the, oh yes, question.
Sorry, Carolyn. Can I ask a question that regarding to your mean specification? Have you controlled, for example, the lab fixed effect in the specification?
Great. Yes, I have a slide on that in the appendix, so I'm glad you asked. Yes. So, so one thing you might think is that just different kinds of labs work on different kinds of proteins. And in fact, we can show you that that's true. So here what I'm showing you is potential on the x-axis and then the university rank of the PI on the y-axis. And so our lower rank is better. So what I'm showing you here is that the sort of people from higher ranked universities are more likely to work on the high potential structures. Now, I look at this and sort of think, if anything, that should actually bias you away from finding this negative quality potential relationship. Because if you think that people from high ranked universities have more resources, you know, maybe it's easier for them to do high quality work. But, you know, you could also think that people are sorting in ways that are harder to observe. Maybe certain people have a taste for quality and they're not willing to engage in these high potential projects because they dislike cutting corners. And it's the people who don't care about quality that are willing to do that. So I think there's many, you know, there's a lot of sorting that could be happening. So I think exactly as you suggest, what we do is we just run these same regressions but include lab fixed effects. And what we find is that actually the coefficients are quite similar when we do that. So here's the figure for potential and maturation. Here it is for potential and quality. And so within a single lab, they are making these kinds of decisions across their portfolio of projects, which I think really rules out this idea that this is all being driven by just different types of people with different preferences for quality sorting into different projects.
Okay.
So yes, no, thank you for asking, because I was prepared for that question. Okay, so, oh yeah, okay, so now we're going to do this structural genomics versus not structural genomics split, but now we're going to look at quality. So again, in blue, we have the university scientists. In red, we have the structural genomics researchers. So as you saw before, there's this negative relationship between potential and quality for the university researchers. It also exists for the structural genomics researchers, but it is far shallower. So how do you think about that? I think that there's sort of two different interpretations that you could have. One is that these structural genomics researchers are less competitive, but they still, maybe for the 20% of projects they publish, there's some element of competition. And so we should expect them to still have a negative slope, but it ought to be shallower. That's one interpretation. Another interpretation is, no, these structural genomics researchers don't care about publishing first at all, but there's some amount of complexity that's correlated with potential that you were not able to control for. And so there's this negative slope that's being driven by that. And so if that's your view of the world, then you think, well, what we should do is take the difference between the blue and the red line. And that's going to tell us how much of this negative slope is being driven by competition. And when you net out the red line, you sort of net out the negative component of the negative slope that's being driven by some sort of omitted variables bias. I think that sort of reasonable people could have differing views. My opinion is probably it's a little bit of both. And so if you prefer to use sort of the difference in slopes as your estimate, that would say that this sort of 90-10 percentile comparison, we lose 0.4 standard deviations of quality instead of 0.7. You know, and my feeling is probably the truth lies somewhere in between those two numbers. Okay, so let me skip this in the interest of time. So now let me get to this point about follow-on work that a few people have alluded to. So In a standard quality ladder type model, researchers could just costlessly build on these rushed lower quality structures. And we would think that sort of from the perspective of scientific progress, it's not bad that people put out this preliminary work in a hasty manner. But in our setting, we think that making a marginal quality improvement really requires resyncing all of the same costs. So in particular, if your experimental data is just not very good, there's no way you're going to be able to build a very good model. And so somebody else who wants to do a better model than you is going to have to go all the way back to this stage of trying to regrow the protein crystal. And the Protein Data Bank has actually sort of run some analyses. They think it costs about $100,000 a structure to redo. So it's expensive. And so I think there's sort of two implications there. It's only going to be worth fixing these particularly bad but important structures. And from the perspective of a social planner, it would have been more efficient to just do it well the first time. And so here we have some results that I think sort of help support this view. So on the left, what I'm showing you is, again, potential on the x-axis. And on the y-axis, I'm showing you the probability that someone came 2 years after or more after the initial structure and redeposited. So I'm trying to cut out the cases where people were just working on it contemporaneously and say, these are cases where someone knew it had already been done, but elected to redo it anyway. And so what you can see is that for these low potential structures, fewer than 20% of them get redeposited. But for these high potential structures, around 80% do. And so that's consistent with this idea that you'll do it if it's worth it. And it's worth it when it's an important problem that has a lot of quality remaining to be eked out. And so that then leads to this second figure. So again, we have potential on the x-axis. Now we have quality on the y-axis. And in blue, we have the quality of the first version of the structure. So this is the figure you've already seen. In red, we have the quality of the best ever version of the structure. And so you can see that a lot of the quality that's lost among these high potential structures ultimately gets recovered when people come along and redo it. And actually, it's sort of comforting to see that at the far right end of this distribution, we end up with basically the same quality as we have on the left-hand side, because that suggests that it's not that these proteins that are high potential just couldn't be done with high quality. Eventually they are. So they could have been done that way the first time. It was just that people were rushing. And what's sort of interesting to see here that the model suggests can happen is we actually end up with this sort of U shape. So the low potential structures, nobody really bothers to redo them, but they're done quite well the first time, so the quality is very high. Then these very high potential structures, while they're done quite badly at first, there's a lot of interest in redoing them, and ultimately those also get done at quite high quality. And it ends up being these structures in the middle that at the end of the day are the lowest quality. And so you can sort of see the two sources here of welfare loss. There's some sort of, quality that we never get in the middle of this U, and then there's all the sort of duplicative costs of resolving these proteins that we wish had just been done well the first time. We think of those as sort of the two sources of inefficiency here.
Carolyn, so
yes.
In terms of that redo of the high potential ones, have you considered the technology progress over time that might make it possible so that it's not the rushing at the first time that make it low quality, but it's just that the frontier is pushed forward?
So that's absolutely possible. But then I would sort of point again to our structural genomics results, which also suggest that in the absence of competition, it doesn't seem to necessarily be the case that that these high potential structures need to be so quality deficient. So I think that your point is very good. The way I sort of think about it is we have like these, 3 or 4 different pieces of evidence. No single one is airtight. I think exactly as you said, there's sort of a very reasonable hole that you could poke in this particular piece, but together, I think it's hard to find one story that sort of rules out all of these sort of complementary pieces of evidence that we have. But yeah, your point is very well taken. So I think like one thing you could try to do is try to do something where you try say something about how much time has elapsed. if a lot of time has elapsed between these blue and red dots, then yes, maybe this has more to do with the technology frontier getting pushed out than it does with somebody just being more careful. And so that's something we could look into.
Carolyn?
Yes.
Yeah, I have a question regarding that graph too. So it's kind of point to, you know, the good side of the competition, right? It's exactly because the first round of the competition generates a very low quality, high potential protein. So people, followers actually has the incentive to improve that, because they can still get a very high citations because the first round is not high quality enough, right? So, that's kind of,
our model suggests that we could have been here in the first round.
Yeah, you're right. So it's kind of a publication game. So you have to leave some space for others, you know, otherwise you will be killed by other scientists.
Yeah.
Yeah, that's a joke. Yeah. Understand. But that's, I just want to point to, you know, I know because at the first round you can reach that high quality even though you have high potential, but then you have to reduce the number of scientists, right? Because you don't need that huge large number of scientists to produce that kind of protein anymore, right? When you reduce the number of scientists, actually you reduce the externalities of the ideas exchange between the scientists. Maybe that's another layer of the welfare you have to consider, right? Yeah.
Yes, absolutely. Okay. So the next thing I'd like to do is just, you know, I've sort of reported to you, oh, we've lost, say, 0.7 standard deviations of quality. But what does that mean? You know, this is sort of this unitless number. And so what I'd like to try to argue is that these effects are big enough to matter for how useful these structures are for somebody who might want to build upon them to develop a drug. So in general, if you ask a structural biologist, you know, how important is it for the to have a really high quality structure, they will say to you, well, it depends what you want to use the protein structure for. So if you just want to use this protein structure to sort of understand how the protein functions inside the body, a very crude structure may be all that you need. But there was one case that was highlighted again and again where you really need a very high quality structure. And that's if you're trying to design a drug that's going to target that protein. And in fact, we were able to sort of find in the literature, there's just these cutoffs where basically, you know, these papers will say if the resolution is not this good or the R-free is not this good, you can't try to design a drug around this protein structure. And in particular, these cutoffs are pretty stringent in the sense that about 40% of the structures in our data don't meet them. So it's not the case that this is sort of just a very low bar. You know, almost half of the protein structures in our data don't meet this threshold. And these quality effects that we're talking about are certainly large enough to move proteins across the threshold from being useful to not useful. And so what we can do actually is just, you know, these are sort of these thresholds were proposed by scientists, but we can actually provide some empirical evidence that they seem to be right. And so the way that we do this is that we're able to actually link protein structures and drugs. So the way that most drugs work is that they are a small molecule that's going to bind to a protein and turn it on or turn it off. And so if you have a really detailed map of what the protein looks like, especially at that binding site, that can give you sort of a clue of what should the shape of the small molecule be that's going to target that protein. And so we take advantage of this data source called Drug Bank that is pretty much a universe of all approved drugs or drugs that are currently undergoing clinical trial. And for every drug, it tells us what is the protein that the drug targets, and it links to the PDB ID or IDs if anyone has solved a model, built a model of that protein. And what we can do is use that to create figures that look like this. And so what I'm showing you here, on the left, I have refinement resolution on the x-axis. On the right, I have R-free on the x-axis. Now, it's a little bit confusing because I wanted to use these raw measures. So the sign is backwards in the sense that as you move to the left in these figures, the quality is improving. And these dashed lines were the proposed cutoffs that I had on this previous slide that were sort of the structural biologists had said you need to be at this level or better in order to be useful for structure-based drug discovery. And the y-axis is the number of drugs that target the structure. So what these figures are saying is that as you move to the left and exceed this quality threshold, it becomes much more likely that you have drugs targeting the protein, which lends credence to these thresholds, because it does appear to be the case that you're much more likely to have a drug targeting the protein if we have models of that protein that exceed these quality thresholds. Okay, so now with the sort of 15 minutes I have left, let me just talk a little bit about welfare. So I think that what is helpful to do is to contrast the scientist's objective to the social planner's objective. And so there's a sort of a lot of notation that I haven't spent a lot of time going through, but let me try to just voice over sort of in plain words what is happening here. So the scientist tries to maximize the probability that they successfully start up the project times their expected private return, which is going to depend on these theta upper bar and theta lower bar weights for winning and losing, minus the investment cost they have to pay. The social planner's objective varies in two ways. The first is that the social planner just cares about the probability that either scientist enters. They don't care which one enters. As long as one of them enters, they're happy. And the social planner's return is not going to depend on these theta upper bars and theta lower bars. The social planner doesn't care who wins or loses. But we do have this extra K term here to capture the fact that the social planner, because of positive spillovers, may value the project more than the individual scientist. So what are the consequences of these two wedges? The first consequence is that the scientist is going to rush relative to what the social planner would like. So they have the same discount rate, but the scientist cares about being first, and the social planner couldn't care less who comes in first. So the social planner wishes the scientist would just ignore this threat of competition, but of course the scientist does not. So that means that the scientist, when left to their own devices, chooses a maturation period that's shorter than what the social planner would like. Somebody else had brought up previously, you know, it's possible the scientist takes too long compared to what the social planner wants. I think what that person had in mind is this idea that the scientist may in fact have a different discount rate than the social planner and may be too patient. So we're sort of not considering that, but I think that's a very reasonable consideration. The second consequence is that if K is very large, so the scientist, the social planner values this project much more than the individual scientist, then investment is going to be too low because this individual scientist is sort of not going to internalize the full value of the project. And then one thing to note is just that if you make these priority rewards equal, so if you set theta upper bar equal to theta lower bar, so you don't get any extra credit for being first, then it becomes, then the scientist chooses the maturation period the social planner would like, because what you've done is you've just totally killed competition in that case. So that's just a helpful fact to note. So when we think about optimal policy here, we're thinking about the policy as being how much credit should we give to the winner versus the loser of the race. And one reason that we focused on this policy is that we saw that several journals had come out with what they were calling a scoop protection policy. So these journals would sort of put out this notice where they say, we at this journal commit to treating papers that have been scooped within, say, 6 months, the same way we would treat it as if it was novel. We will not penalize it. And that really has this flavor of trying to give more credit to the team that comes in second. So what happens in our framework as you make theta upper bar and theta lower bar more equal? It introduces, from the perspective of the social planner, a potential trade-off. On the one hand, as you make these rewards more equal, you increase the maturation time that the scientist is going to choose, and it's going to get closer and closer to the maturation time that the social planner would like. On the other hand, it may also decrease the investment that these individual scientists choose to make. And if we're in a world where K is really large, where the social planner values the project much more than the individual scientist, that's pushing us further and further away from the optimal I. So you're getting closer and closer to the optimal M, but further and further from the optimal I. And so ultimately, what that's going to mean is that the best thing the social planner can do is choose a theta upper bar that's larger than theta lower bar. And so once you have it being the case that the winner is going to get more credit than the loser, you're going to introduce this negative relationship between the project's potential and its quality. So in other words, just because we see this negative relationship doesn't mean we're not at some sort of constrained second best if this is the only lever we're giving the social planner. And Ryan and I actually have another paper where what we try to do is actually just measure what is the reward for being first compared to the reward for being second. And if we take citations as sort of being the measure, we find that that in these sort of cases where we really have a tight race, the winner seems to get about 55% of the total citations and the loser gets about 45%. So we were sort of struck actually by how equitable that appeared to be, how close to 50-50. So we also ran a survey of structural biologists where we posed to them a hypothetical scenario very similar to what we observed in the data. And in our survey, the scientists thought the rewards would be much more unequal. They thought it would be more like a 70-30 split, which I think was just interesting and helps explain, we were struck by how equal these rewards are. Why do people compete so fiercely? Well, if people are sort of miscalibrated and think that the reward to being first is higher than it is, that perhaps can explain why they compete so fiercely. An alternative policy that is sort of suggested by the way our model is set up is that you could just end races early. So in other words, imagine you could have the scientists, once one scientist successfully enters the project, they sink the investment costs and they successfully enter, you could imagine that they just put their hand up and they say, I've entered, nobody else should enter now. And that would fix the maturation distortion if that was honored, because once the scientist has entered, they now know that nobody else is in the race against them, and they can just choose to let the project mature optimally. That's because in the sort of particular way we set up the model, all the uncertainty is in the first step. So once the uncertainty is resolved, there's no benefit to having that second scientist in there. So why don't we just not allow them to be there? The reason I highlight this is that it's just very interesting. In the early days of this field in the 1970s, that was actually basically how things operated. Once a scientist had successfully grown a protein crystal, but before they'd solved the model, they would publish a short paper that just said, I crystallized this protein. And there was a norm in this field that was honored, that everyone else who was maybe working on that same project trying to also grow the crystal, at that point, they just had to go abandon the project. And then that scientist who'd grown the crystal was left to build their model in peace without worrying that they were going to get scooped, and they could sort of do a careful job. So I just think it's very interesting that the field sort of organically adopted a norm that seems to solve this problem and suggests that this model we've developed, that it sort of has these particularities, especially with all the uncertainty being in the beginning, is actually maybe not a bad approximation for how this field actually functions. Ultimately, the field became too big and this norm became hard to sustain. I think when the field was big enough that not all these scientists knew each other, people sort of felt less bound by these social norms. And so that's sort of not how the field functions anymore. And today people do not write papers that say that they've crystallized the protein because they know that that will not be honored and it would just be giving information to their competitors. So that's not how it works anymore, but I just thought it was a very interesting fact that that was how it worked at one point. So let me conclude by just saying that, calibrating the optimal priority rewards, in other words, trying to calibrate this theta upper bar and theta lower bar, I think is beyond the scope of this project. For one reason, I think one thing is just it's very hard to sort of value these marginal quality improvements. We sort of showed you one dimension, this drug discovery, along which they might matter, but these proteins are used for many different things. It's hard to think about sort of trying to holistically value quality. But I think that even more importantly, competition is just impact science in a myriad of ways. And in this project, we've considered one, it might reduce the quality. But there's many other things that we can imagine competition doing. And if you don't sort of consider them all, it's hard to think that you could really say something that sort of what is the optimal amount of competition we want in science. So I think sort of fruitful avenues for future work are things like, how does competition reduce collaboration and the free sharing of ideas? You know, in a very narrow sense, competition seems to speed these individual races up. But if it's also stopping people from talking to each other in a more macro sense, it in fact, sort of ironically, could be slowing science down. Competition may also impact who enters certain fields of science and who is deterred, if some people have more of a taste for competition than others. And that could have important implications for scientific progress. And then I think lastly, this just brings up interesting questions of alternative models of organizing science. And I think that we've seen a bit of a rise of these more collaborative models. So things like the Protein Structure Initiative, which is what generated the Structural Genomics Consortia, the Human Genome Project, which was sort of seen as a huge success and required a lot of coordination and people working together rather than competing for individual glory. And I think a lot of the interest we have in these sort of more collaborative models of science is being born out of this implicit understanding that this competitive model may not purely be good. And so I'll conclude there. I'm happy to take any questions that people have. But yeah, this was wonderful. Thank you so much for having me.
Okay, thank you very much, Carolyn. Thank you very much for the talk. It is very interesting and informative, and I really enjoyed it. And also your time control is great. We already have 7 to 8 minutes for the questions.
Great. And I don't see, is there, is there a chat? Oh yeah, okay, but I haven't missed anything in the chat.
Yeah, in the chat box, we don't have any questions. So for audience, if you have questions, you are welcome to ask.
Hi, Carolyn. Hi, can you hear me?
You're a little broken up. Let me see.
Yeah, can you see me?
Yes.
Yes, I may have a question going really from the very beginning. So, so you are using like structural biology, but I think like, you know, in terms of this field, the structure accuracy may be really important, but, you know, from other field like, you know, in math, right? So there only a difference between who solve this equation and who don't. So this doesn't no such a really you know, like difference between the first and the last, and also in terms of the quality, right? So I may think so what are the implications of, you know, for your wonderful work, you know, for us to understand in other fields, right?
Yeah. So I think that the model helps us think about what are the attributes a field needs to have in order for this problem to matter. And I think one attribute you want is this idea that the projects are close substitutes for each other, because otherwise you're not going to worry too much about getting scooped. So I sort of think about like economics as a bit of a counter example. You can have two people trying to work on the near identical question, but probably ultimately they will write two substantially different papers. And so I think that's a field where getting scooped is, you don't need to worry about it so much. Biology, I think, is different in the sense that, and especially structural biology, because the questions are sort of very narrowly posed. There's an agreed technique you should use, and there's sort of one right answer. And so there, if two people really try to answer the same question, it's very likely that they've created sort of two pieces of knowledge that are just substitutes for each other. And I think that math also has that flavor, as you suggest. There's sort of a proof that's out there, and if two people do it correctly, the second one doesn't add that much value. I mean that's sort of an oversimplification, but maybe to a first approximation, that's right. So from that standard, we might think math suffers from this problem. But then I think the second ingredient you need is this idea that spending more time leads to better quality. And so in structural biology, we think that's the case. If you refine the model more and more, you get a better model. Now in math, I don't know that that's the case. You know, my sort of very simplified view is that you sort of wait for inspiration to strike, but then once you have the sort of key idea, whether you write the proof up over a weekend or you spend 2 weeks really getting the notation just so doesn't add that much value. So in other words, the cost to rushing in math is just not so high. There's sort of not so much quality to lose.
Yes.
So that's sort of my attempt to answer that. Now, I think though, does this generalize at all? Well, I would sort of say that any field where you're running trials, you probably have some element of this, right? Because one decision you have to make is, for example, how many people to recruit for your trial. That makes the process take longer, but you're gonna get a much more statistically powered trial. And so I think that that sort of opens up a whole class of fields where I think this could really be a concern, that basically people run underpowered studies because they just want to get a result out there quickly, and they don't recruit as many people as they should, for example.
Yeah. Yes. Yeah. Thank you so much. Yeah.
Sure.
Yes, Carolyn. Actually, I have a similar question that about the external validity of the findings. Because from my understanding, I think this result will apply to the fields such as structural biology, which pays a lot of attention to the results, to the findings itself, but not to the field such as economics or finance, because we focus more on the I mean that the idea is important, but also the data, the structure, and the method is also very important. And also the review process for the economics papers, for example, is very long. It is very also common for us to have 3 to 4 years for publication.
Yes.
So even if we have a good idea and publish in the MBER working paper, and it is very likely that it will not be published. So do you think that these findings can be applied to the fields such as economics and finance?
I think probably not. But I do think that sort of these two attributes I was just talking about, that the works be substitutable and that spending more time leads to higher quality. I think that is consistent with sort of many subfields of biology. And so you're always speculating a little bit when you're talking about external validity, but my feeling is that probably some elements of this do exist in quite a lot of fields of biology, which I think is, comprises a pretty important subset of science.
Okay, thank you very much. And also, when I read the paper, I found that actually most of the literature, they also focus on the biology, right? So I think this is the reason why they focus on biology.
Yeah.
Okay, thank you. Any questions or comments?
I have a question about the alternative models.
Yes.
Well, we all agree with bad effects or dark side of this our current review tenure system, but it seems I don't see any better alternative. Here you say more collaborative models, but I don't see like very specific suggestion or structure of the alternative.
Yeah, so I think what I mean by maybe the sort of best example of these more collaborative models are the way that in certain parts of high energy physics, the way that they do things there. So my understanding is that people who work at these like large colliders, the way that it works there is that any paper that sort of comes out of that facility is just going to have every person's name on it in some sort of pre-specified order, no matter who does what. And so, that's an interesting thing to think about. On the one hand, I think then you get sort of very good coordination among the people who work there internally. They're not competing against each other. They're sort of going to work together. On the other hand, if you fancy yourself a real physics superstar, are you going to want to go work at this collider where no matter how much of a star you are, your name is just going to appear on the same place in every paper? Maybe not. And so maybe, you know, on the one hand, this maybe leads to more collaboration, less competition, less of this corner cutting. On the other hand, it could also mean that the sort of most talented people are not willing to go work in that model because they don't have a way of signaling that they're really talented. So I think that that sort of is, you know, that's like one example of sort of the trade-off that's introduced. You can sort of shut down some of the problems that maybe I've highlighted in this talk, but I think you're probably also introducing some other problems. Yeah. And so you probably have to think a little, you know, I think the right thing probably is a little bit field specific. And in physics, it also has to do with the fact that the equipment is just so expensive. So they can't have, you know, every person cannot have this for themselves. And so that, I think that has forced this different model, but it's interesting to think about.
Yeah, it's kind of fun, hard to find a way out.
Yes. Yeah. No, and I, you know, if you said to me, I think this is sort of, it's not this first best, but it's the constrained second best. I don't think that that's, I don't know that I would disagree with that. But I also think that it's just interesting if you think about sort of this priority rule, that's just the way science has been organized for hundreds, if not thousands of years. And so is it optimal, or is it just something that we inherited from the generations before us that we could do better? I just think that's a really interesting question. I don't have the answer, but sort of something I think is something that I just think is worth thinking about.
Thank you again, Carolyn, for the very informative talk. And also thank you all the audience for the questions and the comments.